Hi everyone, today we are starting chapter 3 and we're getting into cells, which I of course love because they are the basic unit of life. They are what carry out all the functions that help you survive, all the chemical reactions, all the things that happen inside your cells are essential to you surviving. So, just quick, I'm going to go over the 3 parts of the cell theory. So all things are made of one or more living cells. A cell must come from pre-existing cells, and then a cell is a basic unit of life. So basically, it's the smallest thing that can carry out all the functions of life. So make sure that you have read these pages of the textbook, you've done the review questions, looked over the key concepts here. And then the question of the day, which we'll come back to, is can you drink seawater from the ocean? So I do want to go over this a little bit first. The pre-lecture assignment, you should have gotten this from the reading, but I want to make sure you guys understand the difference between prokaryotic and eukaryotic cells. So prokaryotic cells are typically smaller. They lack organelles, things like endoplasmic reticulum, a nucleus, and an example is bacteria. And I've already filled this in. They lack a nucleus. They do not have a nucleus. They still have DNA. It's just kind of strung throughout the cell. Whereas in eukaryotic cells, it's kind of organized and compacted within this organelle called the nucleus. So eukaryotic cells, like the ones in your body, are typically larger. They do have those membrane-bound organelles. And again, they're found in plants and animals. And they possess or contain or have a nucleus. So something that both eukaryotes and prokaryotes contain is a plasma membrane, or it's sometimes called a cell membrane. And it's like the outer barrier of the cell, and it's a phospholipid bilayer. So if you remember in macromolecules, we talked about phospholipids, which are in the fats or lipids category of the 4 macromolecules. And this membrane is selectively permeable. Meaning that it allows some molecules to enter, but not others. Essentially, it's controlling what can come into and out of the cell. So that's a very important function. And inside the cell, all this stuff floating around here, we have cytoplasm. Which is a mixture of water and cellular molecules, and think of it as having the consistency of Jell-O. So what we call organelles, and what that means literally is little organ. So if you think about the organs in your body, they all have a specific job that they do. The heart is to pump blood, the lungs are to bring oxygen in, your stomach is to digest food. All of these organelles inside the cell have little specific jobs that they do to keep the cell functioning. They're only found in eukaryotic cells, and they are internal compartments with a specialized function. And then make sure you have done the Connect reading quiz for this chapter. And this might seem simple, but these two concepts are so essential for life and almost everything that you talk about in physiology and the function of the body involves some sort of diffusion or osmosis. So diffusion is defined as the movement of molecules from a high concentration gradient to a low concentration gradient. So if you look at this image here, this is like a cell membrane here, a plasma membrane. Got inside the cell. Inside the cell we call intracellular fluid. And outside of the cell we call that extracellular fluid. And inside of the cell, in this picture, these little guys are representing water molecules, and these are representing a particle or some sort of solute. And you can see here that in the extracellular fluid, there are more solutes than there are inside of the cell. So if we assume that these solutes can pass through the plasma membrane, over time what you'll see is that they'll move from outside the cell to inside of the cell until equilibrium is reached. Okay, so they'll go from high concentration to low concentration. And at this point, what students confuse sometimes is equilibrium is that nothing is moving. But you have to remember that molecules and particles are constantly in motion, so that they're still moving back and forth between the cell membrane inside the cell and outside of the cell. But there's no net movement, so there's no change in the concentration. Okay, so just remember diffusion is movement from high to low. This occurs randomly because the movement of particles, and we'll look at this more in just a minute. So next we need to define osmosis, and there's a meme on the internet that I really hate. I should have printed a picture, but it's a person with their head on books, a stack of books, like they're exhausted, and it says something like, "I wish that I could absorb information through osmosis." That bothers me because osmosis is referring specifically to the movement of water. So they should replace that with diffusion. I mean, from high concentration of knowledge in the books to low concentration of knowledge in the head, I guess, I don't know. But osmosis refers only to the movement of water. So anytime you hear the word osmosis, think of water. So we'll define it as the movement of water across a membrane. And osmosis moves from a low solute concentration toward a high solute concentration. And that might seem backwards from diffusion, which you just learned is high to low. But let's talk about this a little more before we get too confused. So in a hypotonic solution, this is a solution with low solutes. It doesn't have a lot of solutes, like I think the picture is here. So think of pure water as being a hypotonic solution. It doesn't have a lot of stuff dissolved in it, and it causes cells to lyse. And the word lyse basically means to burst or explode. So I focus on the O here. Hypotonic means there's a low solute concentration, and then at the same time I also picture the cell. So you've got a cell that's shaped like this, and in a hypotonic solution the water will go into the cell, and the cell will start to expand like a big O, and then could possibly lyse or burst. So that's how I remember it. So let's look at this here. Let's say these are your red blood cells in pure water. So a hypotonic solution, lower solute concentration within the cell. Your cell does have solutes inside, things like glucose and other solutes. And think of this, where can I write this? Water loves solutes. So let's get into chemistry a little bit. And I don't think you necessarily have to know this, but just so you understand the vocabulary I'm using. In a solution, let's say salt water, for example, the solute would be salt, the solvent would be water, and the solution would be the two combined. So water loves solutes. It's going to try to go to wherever the solutes are. So if your cells have more solutes than the solution they're in, water is going to try to get to those solutes and go inside of the cell. And here you can see in this microscopic image, they can swell and possibly burst. And is that a good thing if your cells are lysing? I'm going to say no. Okay, next is a hypertonic solution. This is a solution with high solutes. So I don't know why, but this is how I always remembered it, is someone that's hyper is like high energy. So I just think of hyper as high plus the HY sounds like high. So a solution with high solutes, and this can cause cells to shrivel. So over here in this beaker, we would have a hypertonic solution. So maybe think of something like salt water or glucose here. It's just got a lot of solutes in it. And remember, water loves those solutes, so it's going to want to move to wherever those solutes are. So in this case, they would leave the cell and go to the solution, and the cells would shrivel up. And then again, is that ever a good thing? Probably not. And then last of all, we have an isotonic solution, and this is when the solutes are equal. So in this case, what you will have is water is still moving back and forth between the solution and the cells, but there's no net movement. So it's just kind of in equilibrium. And so this is where your cells like to be in isotonic solutions. They are happy. There's no net water coming into the cell or out of the cell. There's just equal motion of water in and out of the cell, and that's where your cells will stay how they need to stay. So these three terms are very important. So I'm going to circle them. And then it says here, working in pairs, obviously you can do this on your own. Answer the following questions. So define diffusion. This is the random movement of molecules from high to low concentration. So if we were to look at this table below, let's imagine that these are two different solutions and they're separated by a semi-permeable membrane which functions similarly to a cell membrane. So it's going to allow some things to pass through it and some things to not pass through it. So in this case, it doesn't allow these solutes to pass through, and it doesn't specify what those solutes are, and it doesn't really matter, but it does allow water to cross it because water is a smaller molecule. So you're going to hear the word gradient, and that's used a lot in physiology. And what it means is just that there's a difference between one side and the other. So if we look at this one, is there a gradient, yes or no? Well, we would say yes, because there are more solutes here than there are here. Okay, reverse. I got a little ahead of myself. We're just referring to diffusion here and not osmosis. So rewind. This membrane here can allow these particles to pass through. It's semi-permeable. It will allow these molecules to pass through. So, will there be a net movement? And if so, will the solutes move from left to right or right to left, or not have a net movement in either direction? So remember, the definition of diffusion is from high to low concentration. And so you can see here we have a high concentration of solutes on this side and a low on this side. So where is the net movement going to be? From high to low, the net movement is going to be to the left. These solutes will move to the left from higher concentration to low. So here, is there a gradient? Yes, there appear to be more solutes on this side than this side, so yes. And the net movement, it's going to go from high to low, so let's determine which side has the higher solute concentration, and that would be this one. And this one has the lower. So the movement will be from the left to the right. High to low. Diffusion is high to low. Right here, I mean, it's kind of hard to see, but we can go ahead and count. We've got 1, 2, 3, 4, 5 solutes and 1, 2, 3, 4, 5 solutes. So it's equal between both sides. So is there a gradient? No. Their solute concentration is the same. It's isotonic. And so will there be any net movement? No. So we'll say none. But keep in mind that particles are constantly in motion, and so there might be some movement of those solutes among the membrane, but it's going to, in general, stay in equilibrium and have equal amounts of solutes on each side. Okay, so now on to osmosis. The definition is the movement of water, specifically water, that's a key part of the definition, across a selectively permeable membrane. So let's see if we can fill in the table below. So we've got intracellular fluid, which means inside of the cell. Oh, it's already written it there for you. And extracellular fluid, which is outside of the cell. So inside of the cell, we have a 5% salt solution, and outside the cell, we have a 10% salt solution. So the solution, and generally it's referring to the solution that the cell is in, which is this one here. So this one has a higher solute concentration, so we say that it is hypertonic, high for hyper. And will the water move inside of the cell or outside of the cell? Remember that water loves solutes, so if there's a higher concentration of solutes outside of the cell, the water is going to move outside of the cell. And the cells will shrivel. Okay, in this next scenario, we have 87% glucose inside of the cell and 74% glucose outside of the cell. So again, we're referring to the solution outside of the cell. So that would be, it's lower, so hypotonic to the inside of the cell. And remember that water loves solute, so it's going to want to go to where the higher solute concentration is, or in this case it's inside of the cell. And the cells will swell and possibly burst, which we call lyse. And in this third scenario, we have 2% protein inside the cell, 2% protein outside of the cell. That's equal, so we call that isotonic. They're the same. And so water will have no net movement. Because it's equal. So keep in mind, there's still maybe water passing into and out of the cell, but it's happening at the same rate, and so we maintain this equal solute concentration on both sides. So the question of the day was, can you drink salt water, seawater, which is very salty? And I think we all know if we wanted to, we could, but I think a better question would be, should you drink seawater? And so here, what would happen if you did use seawater? And it says use appropriate terminology. So we want to use words like hypertonic, hypotonic, isotonic. And so, again, if you've ever tasted seawater, it's very salty. It has a lot of solutes in it. And so it is hypertonic to your cells. So seawater is hypertonic to your cells. And I have the exact numbers here. Seawater is about 3.5% salt versus 0.9% in your cells. So what it will do, remember water loves solutes, it's going to want to go to wherever the most solutes are, so it's going to want to leave your cells to go to where the salt is, and so that's going to draw water out of your cells. So it draws water out of your cells. And then your kidneys can't get rid of that excess salt. Therefore, you will die of dehydration. So it's better to drink nothing at all than to try to drink seawater because it will take the water out of your cells faster than if you were to drink nothing at all. In addition to that, salt water contains magnesium, which causes diarrhea, which again is going to draw more water out of your body. See if I can spell diarrhea right. It's a hard word for me. There we go. Which will cause dehydration. So, moral of the story, if you're ever lost at sea, don't drink the seawater. It's hypertonic to your cells. It's going to pull water out of your cells to hydrate you even more and cause even more problems. But let's hope that that's a situation that none of us ever end up in. Okay, so on to the plasma membrane. Let's talk a little more about this. This is the outer cell membrane, and it surrounds all types of cells. Prokaryotic, eukaryotic, they all have a cell membrane, and their function is to control what comes in and out. So that's very important. So it's the outer cell membrane. My 7th graders call it the gatekeeper of the cell. It's going to control what comes in or out. And it's the consistency of oil. So even in college, when I was learning about this, I pictured it as like a static thing, like non-moving. It just is how it is. But what's interesting is these proteins that are embedded within it can kind of move around in it. So it's not static. It has the consistency of oil. I think they call it the fluid mosaic model, because it's like this mosaic of proteins inside the phospholipid bilayer that can move around as necessary. So again, it's embedded with proteins, and in this picture, this is a protein here, protein, and then these are your phospholipids. So if you remember anything about phospholipids from a couple lectures ago, they look something like this. So they had that hydrophilic, water-loving head, and the hydrophobic, hopefully you can read that into that picture, hydrophobic tails, so water-fearing. So if there are a bunch of phospholipids and they're put in water, they will naturally orient themselves like this. The hydrophilic heads toward the extracellular fluid and the intracellular fluid because there's water in and out of the cell. And those hydrophobic tails will kind of hide within the middle. So membranes are, again, selectively permeable. Which means they only allow certain molecules to enter and exit the cell. So the question is, what is it going to let in and what is it going to not let in and out? So first of all, it doesn't want to let in ions, which are charged molecules like you see here, and polar molecules, like water, that have a partial charge on one side and the other. So phospholipid is a fat. So remember that fats don't like water, they don't like charged ions, and so they're not going to want to just let those things freely pass in and out. This one is kind of similar, but it's referring specifically to water, which is a polar molecule. And then if you look at this big guy here, that's a macromolecule that we learned about. It's a large molecule. In this picture, it happens to be a protein. So large macromolecules like protein doesn't want to let it in simply because it's huge. Look at it. It's not going to be able to squish through there. So basically these things, they need to get in the cell. We need to have certain ions in our cells. We need to have water coming in and out of the cell. We need to allow proteins to come in and out. And so they need help getting in and out of the cell. So we'll talk about that in a minute. It doesn't just happen through diffusion on its own. But things that the cell will allow to come in and out through diffusion are things that are small, nonpolar. So basically the opposite of these, we want it to be small so it can fit through. We don't want it to have a charge because these hydrophobic tails don't like that. And a couple examples of things that do diffuse across the cell membrane are oxygen gas, because it's so small and it's not charged, as well as CO2, which you'll learn when we get to cellular respiration. It's very important to get oxygen into the cell and CO2 out. And also steroids. So how do we get these molecules into the cell that can't just diffuse across the cell membrane? So passage of non-permeable molecules through membranes. They need help getting in through these transport proteins that we saw that kind of move around within the phospholipid bilayer. So facilitated transport sometimes called facilitated diffusion. And if you think of the word facilitate, if you are facilitating somebody, you are helping them. And so molecules move via diffusion through protein channels in the membrane. So they're still going from higher concentration to lower concentration, but they have to go through these specific proteins to get there. So the proteins are helping them get inside the cell. So on this picture, if you look here, we have a higher concentration outside the cell, so I'm going to call that hypertonic. If you can read my writing there. And then inside of the cell, it's hypotonic. I guess this picture isn't really illustrating what I'm wanting it to, because in that case, oh no, that's right, it's going from high solute concentration to low, and the proteins will help it get inside of the cell. I get confused when I'm talking about diffusion versus osmosis. So right now we're just talking about diffusion. So it's the movement of molecules. Okay, another way that we can get these molecules that can't go through the phospholipid bilayer in and out of the cell is called active transport. Now this is not a form of diffusion, because if you look, it says the movement of a solute from low to high concentration. So if you think about it, and the way that I think about it is I've always pictured a slide. So if you're going down a slide from high to low, that's just like, whee! You don't require any energy, you just kind of go. from high to low. But if you were going to go in the reverse from low to high and climb up the slide, that's going to take a lot of energy. And we call that going against the concentration gradient. So the type of energy that your cells use to do this is my good old ATP. Remember, the adenosine triphosphate is what that stands for. So you've got an adenosine, those 3 phosphates, and between the second and third phosphate is that high energy bond that releases energy. And that's like the currency that your cells can use to do work. So that's just one example of where ATP is used, is to pump these solutes against their concentration gradient from low concentration to high concentration, which is not the natural motion of molecules. And again, it requires energy in the form of ATP. So in this picture here, we've got a protein pump, and it's taking ATP energy. That third phosphate is breaking off to form ADP, adenosine diphosphate, where you only have the 2 phosphates. And it is pumping, let's see what we've got here. It's pumping potassium ions against their concentration gradient, so from low to high. And then it's pumping sodium ions against their concentration gradient from low to high. And again, if you're climbing up a slide from low to high, it requires energy, just like active transport. Okay, this stuff becomes very important in physiology. Hopefully now you just understand the basics that if you're going to go from low to high, that's not the natural motion of molecules, it takes energy. But if you're going from high to low, that is the natural movement of molecules to just even themselves out. And that doesn't take energy, but if it can't cross directly through the phospholipid bilayer, it needs some help from a protein, and that's facilitated transport or facilitated diffusion. All right, this last page for the day is important to know and understand, but there are only a certain amount of things that we can test you on, and we've covered so many things so far. I will give you a hint and tell you you will not be tested on this, so you might want to make a note of that somewhere, but it still is important to know, so I'm going to cover it. So bulk transport, if you think of something in bulk, it's large volumes. So it's taking large volumes of something inside the cell by forming a vesicle with the plasma membrane. So a vesicle, if you look at this is the phospholipid bilayer, the plasma membrane, and it just kind of, I think the word is invaginates. It kind of invaginates in, it forms this cave going in, and then the plasma membrane ends up surrounding the vesicle and whatever you just brought in, and then it can take it to wherever it needs to go in the cell. So there are 2 types of this. There is phagocytosis and pinocytosis. So we'll start by looking at phagocytosis. This literally means cell eating. Anytime you see cyto, that means cell, and phago means eating, so cell eating. This is when it brings in large foreign matter, like you see here. So where would this happen in your body? Why is this important? This happens in your immune system. So your white blood cells can swallow, they can eat things, foreign matter like bacteria, and then once it gets inside, it will digest it and get rid of it, essentially. So very important for your body. So white blood cells. Swallow foreign matter, like bacteria. So it kind of reminds me of a Pac-Man. It just opens its mouth, surrounds it, closes off, pinches in, and there you go, inside the cell. Penocytosis means cell drinking. So an example of this would be in your small intestines, and your small intestines are lined, it needs to have a great surface area to absorb things. So they're lined with these little finger-like projections called microvilli. Sorry, I'm a teacher. I'm used to talking with my hands, and so I'm trying to get my hands in here as much as I can because I can't see face to face. But anyway, to grab water and solutes and bring them inside the cell. Or to bring them into, the cells. So example, villi and microvilli in the small intestine during digestion. Grab water and solutes and bring them in. I don't know if you can hear that. That's just my pants squeaking against my chair. I'm sorry. Okay, so penocytosis is nonspecific, meaning it's just going to grab and bring in whatever's out there. So again, it means cell drinking, so it's bringing in water and whatever solutes are in there with it. So same type of process, you have the cell membrane, it starts to invaginate and pinch it off there, and it'll just grab water and whatever solutes are there, close it off, surround it with that phospholipid bilayer, and now you've got a vesicle that can move around the cell and go wherever it needs to go. And this third one is called receptor-mediated endocytosis. So endo means within, cytosis means the cell, so it's bringing things within the cell. So this is when solutes bind to receptors on the membrane and all are brought in at once. So you can see here there are these receptor proteins embedded in the phospholipid bilayer, and they attract certain specific solutes that will stick to it, and then it will pinch off, go inside the cell, and that's how you get those solutes inside of the cell. So solutes bind to receptors on membrane, and all are brought in at once. This is how cells acquire cholesterol. So example, iron receptors in your red blood cells. Anytime you see RBC in biology or physiology, it's red blood cell. If you see WBC, it's white blood cell. But it's very important to get iron into your red blood cells, and so your red blood cells will have these receptor proteins specifically for iron in their cell membrane to get the iron to attach here and then get brought into the red blood cell. And then there's one last note here, specific. In that specific solutes bind to receptors like lock and key. Only certain keys fit in certain locks, and only certain solutes will bind to these specific receptor proteins. So, in summary here, let's make sure we know about diffusion and osmosis. So, diffusion, and we already essentially wrote this down, but I just want to make sure you understand, is the movement of solutes from high to low concentration. So if we have a beaker here and it's separated with two different solutions, we've got, let's say we've got salt water on this side and we've got pure water on this side with no solutes in it. This side would be hypertonic because it has a high level of solutes. This side would be hypotonic because it has a low level of solutes. So assuming that these solutes can pass through this membrane here, there should be a movement from high to low. And then what we would end up with over time is equilibrium would be reached. How many do I have? 1, 2, 3, 4, 5, 6, 7, 8. And eventually we would end up with isotonic solutions on both sides. Alright, so think of all things wanting to be equal in diffusion. From high to low, it doesn't require input of energy because it's like going down the slide from high to low. And the solutes will tend to equal out. Osmosis is specifically the diffusion of water across a selectively permeable membrane so it's going to let certain things in and not let others in. So let's see if I can not make this confusing. So let's do the same situation, but this membrane here is selectively permeable. So here, I'm not showing the water molecules, but it does let water molecules freely pass, but it doesn't let the solutes freely pass. Just imagine that the solutes are too big and the pores are too small. Water is a relatively small molecule, so it can pass through, but the solutes can't. And so with osmosis, what would happen is we would call this the hypertonic solution. It has more solutes. This would be a hypotonic solution. And so remember, water loves solutes. So this time we're referring to the movement of water, not the solute like we were up here. And so the water is going to want to move toward the solute from low solute concentration to high solute concentration. And the water would move in this direction. The key is these solutes have to be trapped. They can't be able to cross through here, otherwise they would just move over through diffusion and equal things out. So they're trapped over here, but water can move through, so in this case water would move over to this side. So a real life example of this, if you think about plant cells, of course osmosis is important for plants. If you have celery and it is limp and flimsy and you stick it in water, what will happen is because of osmosis, the celery itself has a lot of solutes inside. basically cellulose, so carbohydrates and sugars, and the water that you put it in is hypotonic, and so that water is going to be attracted to the solutes inside the celery, so it's going to go into the plant cells, and that piece of celery will become rigid. Is that the word I'm looking for? Rigid, stiff, not so bendy, not so flimsy, not so limp. Okay, I think that's it for today. That's part 1 of cells, and we'll be back with part 2 next time. Please, if you are confused or have any questions, send me a message on Canvas. I would love to help you. I want to make sure everyone's caught up and understanding, so if there's something you're confused on, let me know. We can walk through it through the messages, or you can even set up a Zoom or Google Meet call with me. So, yeah. Have a great day. Thanks, guys.